Mimex Tissue Model Systems is a culturing platform for generating sustainable, accessible, and physiologically relevant 3D organ tissue, optimized for the gastrointestinal tract, respiratory system, and other epithelial organs. Tissue generated using this technology can be utilized for drug discovery, toxicology screening, biomarker discovery, and disease modeling. Mimex tissue model systems harness the power of adult ground state stem cells located within epithelial organs. When isolated and cultured, an individual tissue-specific stem cell clone can be rapidly expanded and easily maintained in culture. Switching these cells into an air-liquid interface system induces the stem cells to differentiate into three-dimensional ex vivo tissue that mimics the native organ. For example, examining colon-generated tissue using the Mimex GI system, we see expected cytoarchitectural features, such as intestinal crypts and apical to basolateral polarity as indicated by villen expression. The tissue also contains goblet cells, Paneth cells, and enteroendocrine cells. The power and benefits of Mimex tissue models is rooted in the unique characteristics of adult ground state stem cells. These cells, when isolated from an organ or subregion of an organ, can be expanded in culture and will differentiate back into functional region-specific tissue. This specificity extends into disease modeling, where stem cells isolated from pathogenic tissue will differentiate back into the diseased tissue ex vivo. Individual clones of adult stem cells are also easily isolated during expansion in Mimex reagents. Gene editing techniques can be applied to these clones to differentiate a sustainable source of genetically modified tissue. Mimex tissue model systems build upon the physiological and structural advancements of existing 3D models, such as organoids, by providing better accessibility and lower variability across experiments. Unlike organoid and spheroid models, the Mimex system generates 3D tissue on a 2D interface, where the apical or luminal surface of the epithelium is accessible. This added access to the functional surface of the tissue makes Mimex more available for experimentation. Tissue generated using Mimex reagents arises from a monolayer of stem cells. Because of this, the size and shape of the tissue is determined by the dimension in which the monolayer of stem cells is plated. This consistency translates into low variability across wells, making this system ideal for high-throughput studies. Mimex tissue models are designed to bring accessibility, consistency, and flexibility into 3D cell culture research. By offering a full set of reagents to isolate, expand, and differentiate tissue-specific stem cells, the Mimex system is primed to transform the use of 3D tissue modeling for drug discovery, toxicology screening, regenerative medicine, biomarker discovery, and disease modeling. Interested in Mimex tissue model systems? Visit our website, rndsystems.com/Mimex.